Hello students, again today we are going to study one more point from the third topic, that is inheritance and variation, and the point is linkage and crossing over. Let us see first point, that is linkage. It is a known fact that several genes are present on the chromosome, as chromosomes are carriers of heredity. These genes have tendency to be inherited together. Such genes are called as linked genes. This tendency of two or more genes present on the same chromosomes that are inherited together is known as linkage. So those genes get inherited together are known as linkage. So this linkage concept was first time discovered by Batson and Punnett in plants, and in animals by T. H. Morgan. So linkage is of two kinds of linkage. First one is complete linkage and the second one is incomplete linkage. So in simple language, you can see genes are present on the chromosomes, and these genes get inherited from one generation to the another generation is and these genes get inherited from one generation to the another generation together, and this phenomenon is known as a linkage, and these genes are known as linked gene. Let us see the complete linkage. The linked genes which are closely located on the chromosomes do not separate. There is no crossing over. Due to the no crossing over and inherit together, they are called as these are called as completely linked. These are strongly linked genes, and the phenomenon of their inheritance is called as complete linkage. While incomplete linkage, the linked genes which are distantly located on the chromosome and have chance to segregate, okay, by crossing over, and this is known as incomplete linkage. So these two terms are opposite to each other. Okay, so this thing is found in the X chromosome of Drosophila males show incomplete linkage. Let us see the most important thing, that is the sex linkage. As I said earlier, there are six chromosomes found in human being as well as some other animals, and these chromosomes are X chromosome and the Y chromosome, because these two chromosomes determine the sex in the new offsprings. Okay, so these two chromosomes are known as sex chromosome. So sex linkage, we have studied the linkage thing. Now just see the sex linkage, the transmission of X linked and Y linked genes from parents to offspring is called as sex linked inheritance. So the transmission of X-linked and Y-linked genes from parents to offspring is called as the X sex-linked inheritance. Sex-linked inheritance is of three types. Yes, there are three types of sex linkage: X-linked, Y-linked, and combined X and Y-linked linkage. So sex linkage is of two kinds. Complete sex linkage. Let us see first one, that is the complete sex linkage. These genes are located on the non-homologous regions of the X and Y chromosomes, and they inherit together because crossing over does not occurs in these regions. For example, color blindness. Okay, now just see one more thing, that is the incomplete linkage. It is exhibited by genes located on the homologous regions of the X and the Y chromosome, and they do not inherit together because of crossing over. Crossing over occurs in this region. Okay, so these genes are basically, you have studied already homologous and the non-homologous regions of the X and the Y chromosomes. So basically, so these genes are located on the homologous region of the X and the Y chromosomes, and they do not inherit together because of the crossing over takes place in these regions. So these uh, genes are known as in this type of phenomenon is known as incomplete sex linkage. For example, X and Y linked traits are total color blindness, nephritis, uh, retinitis pigmentosa, etc. Okay, so these are few examples of the incomplete sex linkage. Now just move towards the most important point, that is the crossing over. So crossing over, this is most important event happens in the uh, recombination. This is very very essential for the recombination, that is crossing over. Okay, so crossing over is a process that produces new combinations. As I said earlier, this is recombination. New combination means the recombination. So crossing over is a process that produces new combinations or recombinations of genes by interchanging and exchanging of corresponding segments between the non-sister chromatids of the homologous chromosome. And this thing occurs during the patching of the prophase 1 of meiosis. Okay, so this particular thing, that is crossing over, takes place between the non-sister chromatids of the homologous chromosome during the meiosis, and the phase is prophase 1 and the subphase is patching. So this prophase involves different subphases, that is the leptotin, zygotin, patchotin, diplotin, and the dikinesis. So this thing, that is the crossing over, takes place during the patchotin of the prophase 1 of meiosis first. Okay, so the term crossing over was first time coined by the Morgan. The mechanism of crossing over, this mechanism of the crossing over takes place, as I said earlier, during the meiosis first, and the surface is prophase 1 of the prophase 1, and this prophase 1 involves different subphases again, that is the, as I said earlier, leptotin, zygotin, patchotin, diplotin, and the dikinesis. So this crossing over takes place only in the patchotin, in which the non-sister chromatids exchange their genetic material, that is, you have studied already, genes by the interchanging and exchanging the corresponding segments between the non-sister chromatids of the homologous chromosome. So this thing takes place between the non-sister chromatids of the homologous chromosome. Sorry. Let us see the crossing over concept and the linkage concept with the help of the Morgan's experiment. Okay, so let us see Morgan's experiments showing linkage and the crossing over. Morgan carried out experiment on a Drosophila melanogaster. It is commonly known as a fruit fly because it can be easily cultured. That Drosophila can be easily cultured. Be cultured. Morgan carried out dihybrid process in Drosophila to study the sex-linked inheritance. Morgan crossed yellow-bodied, white-eyed female to wild type, that is brown-bodied, red-eyed male, and intercrossed their F1 progeny. He found deviation to the Mendel's ratio, that is 9:3:1. So Morgan and his co-workers found those genes are strongly linked. They show very few recombinations. Okay, let us see the example. Is in this combination, in this first one, in this first one, Morgan and his co-worker found the genes are strongly uh, linked on the chromosomes. They show, because of this reason, they show uh, a recombination, but that recombination was very less, that is 1.3% in this particular cross. While in this cross, the recombination was 37.2%. Okay, so this is simply regarding the difference between the recombination in the first cross and the recombination in the second cross. So In this first one, Morgan crossed yellow, yes, Morgan crossed yellow-bodied, white-eyed female. This is yellow-bodied, white-eyed female with this one wild type, that is the brown-bodied, red-eyed male. Okay, and he found in this cross he found the recombination was only 1.3%, because in these in this particular cross the genes were tightly located on the chromosomes and they unable to uh, cross over. So because of this reason the recombination was very less, that is 1.3%. And uh, in this cross the recombination was 37.2%, because the genes were loosely arranged on the chromosomes. So crossing over was more possible in the second cross. Okay, so the recombination rate was 37.2%. Okay, so they show very few recombination in this particular cross, that is 1.3%. And first cross shows crossing over between the Y and the W, and the second cross shows crossing over the between the W and the M gene, and the dominant alleles are represented by plus symbol. So this was simply regarding the Morgan's experiment of linkage and crossing over. Okay, let us move towards the one more point, that is the autosomal inheritance. As I said earlier, there are two types of chromosomes found in human being: autosomes and the sex chromosomes. Out of 46 chromosomes, means 23 pairs of chromosomes, 22 pairs of chromosomes are autosomes and one pair of chromosomes is sex chromosomes. Okay, so there are two sex chromosomes, whether XX or the XY, and 22 pairs of chromosomes are autosomes. So this inheritance is simply related to the autosomes, means this is related to the 22 pairs of the chromosomes. Now just see the basic things regarding this particular autosomal inheritance. Human somatic cell, that is 2N, it is diploid cell. 2 cell contains 23 pairs of chromosomes. Okay, as I said earlier, there are 23 pairs of chromosomes. Out, out of 23 pairs of chromosomes, 22 pairs are autosomes and one pair is sex chromosome. So they can be divided functionally as autosomes and the sex chromosomes. Single pair of chromosome is involved in the sex determination and the remaining 22 pairs are called autosomes. Autosomes control a variety of traits other than the sex. These traits are called autosome linked traits. Transmission of body characters other than sex linked traits from parents to their offspring through autosomes is called as autosomal inheritance. So as I said earlier, chromosomes transmit some characters from one generation to the next generation. So that is called as the autosomal inheritance. Autosomal inheritance. Okay, let us see first example of the autosomal inheritance, that is widow's peak. A prominent V-shaped hairline on a forehead is described as widow's peak. So V-shaped hairline in these two diagrams. This first diagram shows a V-shaped hairline on a forehead. So this V-shaped hairline is known as a widow's peak. It is autosomal inheritance. While in this diagram or in this figure, you can see a straight hairline on the forehead. Okay. Now just see, widow's peak occurs in homozygous dominant, that is W and W, and it is also in the heterozygous, that is capital W or the dominant W and the recessive W individuals. Individuals with homozygous recessive, that is small w and the small w, or recessive W and the recessive W genotype, have a straight hairline. There is no widow's peak on the forehead. So both males and females have equal chance of the inheritance. So the gene responsible for this widow's peak is a dominant W gene. So both males and females have equal chance of the uh, this widow's peak inheritance. Now in this condition, in this inheritance, this homozygous that is with dominant W and the dominant W, and in heterozygous, that is dominant W and the recessive W, this is heterozygous. Okay, so both these individuals show V-shaped hairline, that is widow's peak on the forehead, while the homozygous, I repeat, homozygous recessive with a genotype, that is recessive W and the recessive W genotype offspring, shows a straight hairline. There is no widow's peak on the forehead. So this individual shows a straight hairline on the forehead. So this is first autosomal inheritance. First example of the autosomal inheritance, that is widow's peak. Let us move towards one more autosomal inheritance, that is phenylketonuria. Phenylketonuria, it is an inborn metabolic disorder caused due to a recessive autosomal gene. So again, this uh, disorder is mainly found in some people due to the autosomal gene. Okay, so this is phenylketonuria. So it is, as I said earlier, it is inborn metabolic disorder. This is disorder caused due to the recessive autosomal, again this is a recessive autosomal inheritance. Okay, the gene is a recessive autosomal gene. When the recessive genes are present on the homologous condition, phenylketonuria, phenyl phenylalanine phenylalanine hydroxylase enzyme is not produced. Now you see, the most important thing in this inheritance, autosomal inheritance, the gene responsible for this is a recessive autosomal gene. When a recessive genes are present in homozygous condition, phenylalanine alanine hydroxylase enzyme is not produced. In this homozygous condition, this enzyme, that is phenylalanine hydroxylase enzyme, is not produced. This enzyme is essential, and this enzyme is essential for the conversion of amino acids, amino acid phenylalanine into the tyrosine. This enzyme, that is phenylalanine hydroxylase enzyme, is essential for the conversion of amino acid phenylalanine into the tyrosine. Due to the absence of this particular enzyme, phenylalanine is not converted into the tyrosine. Hence, phenylalanine and its derivatives are accumulated in blood and the cerebrospinal fluid. So, due to the deficiency of this particular enzyme, this enzyme is not produced due to the presence of recessive gene on the in the homozygous condition. So, when recessive genes are present in the homozygous condition, phenylalanine phenylalanine hydroxy enzyme is not produced. This enzyme is very essential for the conversion of amino acid phenylalanine into the tyrosine. So due to the absence of this enzyme, phenylalanine is not converted into the tyrosine, and it gets accumulated in the blood and the cerebrospinal fluid. This affects development of the brain, yes. Then it causes the mental retardation. Excess phenylalanine is exerted in the urine, sorry, excreted. This excess phenylalanine is excreted in the urine. Okay, and the disease is called as phenylketonuria. So this is called as phenylketonuria. So in So the gene responsible for this is a recessive autosomal recessive gene, and when this gene is present in the homozygous condition, not in the heterozygous condition, this is recessive gene, and when this gene is present in the homozygous condition on the chromosome, this enzyme, that is phenylalanine hydroxylase enzyme, is not produced, and this enzyme is very essential for the conversion of the phenyl amino acid phenylalanine into the tyrosine. Okay, so this conversion is essential, otherwise this absence of this phenylalanine is not converted into tyrosine, and hence phenylalanine and its derivatives are accumulated in the blood and the cerebrospinal fluid, and this affects the development of the brain and causes the mental retardation, and the excess phenylalanine is excreted in the urine as a excretory product, and this is that's why it is called as a phenylketonuria. Okay, so this is simply regarding the autosomal inheritance, that is phenylketonuria.